Today we're going to talk about cell structure 1. We'll do basically three things. We'll look at the different types of microscopy, three different types of way of looking at cells. We use the toluidine blue. We will also look at H&E preparation, as well as we will look at PAS staining, which stains for carbohydrate rich. In addition to that, we'll look for ultrastructural changes that we see at the at the electron microscopic level. Here we see the atlas view of the intestinal absorptive cells lining the intestinal villi, as we see at the top and through here. These are the intestinal absorptive cells lining this intestinal villus, which is located in the small intestines. Here we can see the small intestines with the villi projecting up through there, and intestinal absorptive cells can be seen through here, the pink that we see through there. The light components we see there are actually goblet cells. So we have cells that line the lumen up through here, the intestinal absorptive cells. There's also connective tissue cells in through here, smooth muscle cells in through there, which is responsible for paracelsus to occur. This is one of our slides, 148, which is a human ileum, and you can see the connective tissue band in through there, as well as the intestinal absorptive cells lining the lumen. So the intestinal absorptive cells, this epithelium line viscera and the line cavity, so they line this gut. So we're working in this area right here is what we see in the intestinal absorptive cells. As you get down toward the base of these cells, you can see there's other cells that have a different type of characteristic of staining. So we have the lumen, the outside of the gut, the muscle layer of the gut, the connective tissue of the gut, and the epithelium lining the surface of the gut. So here we can see again more closer view. We got the connective tissue core in through here, epithelium on the surface, and this is the epithelium absorptive cells located. These are simple columnar cells, column shapes, long nuclei in these cells, as we can see around through each one of these intestinal villi has a series of intestinal absorptive cells which have intermittent, they have goblet cells in there as well. 148, the ileum, a little closer magnification, we can see these intestinal absorptive cells and the goblet cells. We can see the nuclei of these cells kind of lined up. So these are simple columnar cells. We can see them right here. There's a nuclei, and there is a brush border that we see associated with the luminal surface. Nuclei, cytoplasm, columnar cells, connective tissue, muscle layer in through there, and more connective tissue, as we see. At the base of these cells, you also have other type of cells. These are the Paneth cells. The Paneth cells secrete enzyme to kill bacteria that go in through there, and we'll learn about those later on. But right now we're concentrated on intestinal absorptive cells located right in through there. And here we can see throughout, we can see little mitotic activity. These are metaphase plates associated with the production of new cells. A little higher magnification shows you the intestinal absorptive cell. Indeed, it is a column. As we see there, this is the nucleus. Nuclei are more than one cell. There's a goblet cell, and there's the brush border. You can see this kind of little fuzzy thing right in through there, or glassy looking thing. That's the brush border, and that goes all around the cells that we see in through there. Brush border continues. There's no brush border over the goblet cell as we see it discharge in through there. Next we can see the PAS staining. If you stain goblet cells, they are rich in carbohydrates, and so we can see that goblet cell, the mucus inside the cell, stains carbohydrate rich. Also, the brush border along the outside of them, intestinal absorptive cells going from here to here, these are the tall columnar cells, the brush border on the luminal surface is also PAS positive. In addition to that, the basement membrane that the intestinal absorptive cell sits on also is PAS positive. And we can see that this is a stain to slide 249 with PAS staining, periodic acid shift staining, and it shows you the goblet cells, which has a lot of carbohydrates inside there, and it also shows you the PAS positive brush border surrounding each of these intestinal absorptive cells. Intestinal absorptive cells run from here to there. So these are individual cells, and then these are nuclei of the individual cells in through there. So again, we can see the brush border and the goblet cells. Goblet cells, brush border is highly PAS positive. In addition to the brush border, as we see here, and the goblet cells are PAS positive, also right in through here you can see the basement membrane of the intestinal absorptive cells is also PAS positive. So this is connective tissue in through here. This is the lumen, goblet cells discharging their contents, and a brush border is PAS positive in these cells. So we looked at the small intestines. Now if we look at the large intestine, we can see that you also have intestinal absorptive cells, but you have a higher concentration of goblet cells. Also, you don't have villi, you just have intestinal crypts. So here we see the brush border on the intestinal absorptive cell. This is one intestinal absorptive cell right there, columnar shape, more cytoplasm above the nucleus and between nuclei. And then these guys are goblet cells, some of which you might be able to see them discharge their contents like this one. So here we see the intestinal walls of the intestinal absorptive cell, brush border on the surface. Right in here we can see the terminal bars. The terminal bars are where adjacent cells are attached together with junctions. Here's a higher magnification showing you the intestinal absorptive cell, the brush border, the goblet cells in through there, the nuclei of the intestinal absorptive cells. There's largely light staining, and you can see a distinct nucleolus. You can see a nucleolus within, here we can see more nucleoli within the intestinal absorptive cells. Also in through here you can see plasma cells. Plasma cells produce antibodies. So this is a lumen of the things that come in that you eat, the ingesta that comes in through here. You need to have plasma cells to produce antibodies to counter any antigens that might be coming in. These are smooth muscle cells that are in through here. These guys here with a lot of cytoplasm around them. And oftentimes you see a kind of cartwheel characteristic of these cells, these plasma cells that we see in through there. Here again is another view of these. This is the base of these cells. We don't have the pennant cells that we saw in the small intestine. Here's a lymphatic with a lot of white blood cells. These are white blood cells in through here. These capillaries here are being cleared of blood, but these are lymphatics. Lymph fluid. These are fat cells in through there. Little bit of smooth muscle going around through there. Connective tissue with lots of plasma cells in through there. Another type of stain that was H&E preparation that we're seeing. This is toluidine blue. Remember, with toluidine blue, everything is blue. You have to look at the size of things, the shape of things, and the intensity of staining. Here's a lymphatic through the center. This is a centrolacteal. These are the intestinal absorptive cells in through there. These are their nuclei, and there is the brush border. A little higher mag, we can see the brush border running through here, all the way in through there. So this would be an individual intestinal absorptive cell right in through there. This is the nuclei of those cells. Again, we can see it a little higher magnification. So this would be an individual cell that will go in through here, columnar cell. These are taller ones as you can see right here. These are the nuclei of these cells, the brush border, This is the secretions of the goblet cells that are located in there. Central lacteal, as we see through there, there's places here where there would be blood vessels are running through. There's a capillary right in through there. Here we can see another one. Again, we can see the brush border projecting up. Individual columnar cells, more cytoplasm above the nucleus and between nuclei, so a simple columnar epithelium lining these. Now in the connective tissue here, we can see the central lacteal, which is what we saw before, which is the lymphatic, drains the lipid that you absorb in your gut. And here we see some mast cells. These are mast cells, a certain type of white blood cell. This is another slide of rat intestine showing you the outer muscle wall, connective tissue there, and also we see a host of intestinal absorptive cells that actually cut a little odd at this shape. They're cut along the height of the cell as opposed to along the length of the cell. But here we might be able to see some that are cut along the length of a cell, goblet cell, intestinal absorptive cell, and here we can see the same thing. These are intestinal absorptive cells in through there with connective tissue down below. These darker tissue right in through there is smooth muscle, and this is smooth muscle there. Large blood vessels in through there, connective tissue in through here, and intestinal absorptive cells lining the lumen. A higher magnification shows you the brush border of these intestinal cells, and you will see the goblet cells discharging their contents in the lumen. So these are intestinal absorptive cells, these are goblet cells. Now, unlike the other specimen with H&E, mitochondria can be seen. These clumps of clusters of cells, organelles right here are actually mitochondria. So these are mitochondria at the tip. Mitochondria are needed for absorption of food stuff, for ATP production, for transport of things in the lumen. And here again we can see the little higher magnified clusters of mitochondria. Here we can see little clusters of mitochondria cut through the apex of the cell. So this is basically a columnar cell, one column, another column. You can see the nucleus is largely euchromatic, light staining, and you see the nucleoli. And around the edge you can see some heterochromata, which is a dark staining chromatin material. So we can see brush border right in through there, height of the cell, nuclei, mitochondrial clusters. In a certain place you can see the cluster. Also, some places you might be able to see the junction between adjacent cells. In this case, you can see the junctions going through there around multiple cells. It's cut along the junction, and again you can see it here. Here you can see the terminal bars, but here they're continuous because indeed they form a belt around the cells. If we look at electron microscopy at the base of the cell, so this is connective tissue with collagen, and there's a basal lamina. The epithelium, which is this epithelium cell, intestinal absorptive cell, is an epithelial cell. It sits on the basement membrane. At the electron microscopic level, you can actually see part of that basement membrane as the basal lamina. And that's that fuzzy coat that we see right in through there. It's at the base of all epithelial cells. But here we can see individual cells, the base of the cell, lots of mitochondria. So this is one cell right in through there, and here's its nucleus. And the nucleus has euchromatin, which is light staining, and then on the edge we see heterochromatin. And sometimes you might be able to see nuclear pores. But these are all mitochondria of different cells. So the host of different cells there, mitochondria at the base for transport at the bottom. Also, we can see this is the luminal surface of the cell. We can see the brush border. And the brush border is really microvilli. Uniform microvilli of high density is a brush border. In contrast, the goblet cell that we see here has microvilli on its surface, but they're not in high density, so they do not make a brush border. But they are carbohydrate rich because the secretions themselves are carbohydrate rich. Also, the brush border is carbohydrate rich because it has a glycocalyx coat that surrounds it. There's also mitochondria located in the apex, and here we see junction between adjacent cells, the occluding junctions. Lipid inside the cell, mitochondria, the membrane, and we want to see a kind of fuzzy outside, which is the brush border. If we see a higher magnification, we can see the mitochondria. There's one there, another one there, another one. You can see the cristae, nice cristae in these mitochondria. You can see a junctional complexes. These are the tight junctions between two adjacent cells. Another junction in through there with desmosomes along the way, but these are the occluding junctions, so-called zonia occludens. And also you might note that these microvilli have microfilaments in through there. So the microfilaments is actually what pushes the plasma membrane out and makes the microvilli. If you come down from the surface of a cell a little, you see a lot of Golgi apparatuses. One Golgi, there's another Golgi, another Golgi, another Golgi. A Golgi, it looks like a series of pancakes that's been cut. A series of flattened vesicles is what we see with the Golgi apparatus. And these are membranes between two cells. This is actually two membranes, one from that cell and one from this cell. Is what we can see. Also, you can see other structures in through here. You can see the nucleus with a euchromatin and heterochromatin, as we see right there. Heterochromatin and euchromatin is a light staining, is euchromatin. Heterochromatin is a dark staining. Another Golgi apparatus there. We can also see the rough endoplasmic reticulum and a little bit of budding a rough endoplasmic reticulum, and here we see some coated vesicles. So today we talked about cell structure 1, we talked about electron microscopy, as well as different types of visualization with a light microscope. With toluidine blue, where you looked at sizes of things and density of stain and the shapes of things, and we'll look at H&E, which is pink and blue, and then we looked at PAS staining of the tissue.